ready to review those materials to be ready for the exam. And so T-cell activation, we'll be able to spend a little bit more time on this now that I'm not going to try and jump into the next chapter. It's pretty important to understand this topic. Understanding this is going to come into play when you're thinking about vaccines, you're thinking about immune responses to pathogens, to autoimmune disorders. So understanding kind of the logistics of how you activate a T cell, because again, we have negative and positive selection happening as T cells are developing, and then they will get out into the periphery. But for the most part, T cells don't recognize self-antigen, but we still have mechanisms in place to protect against auto-antigen specific T cells being activated. And so T cells, there's still some checks and balances going on out in the periphery. And so T cells, once they leave the thymus, they're in this naive state. So again, naive means that they have not encountered the antigens yet. They are a mature cell in that their T cell receptor is already established, and they are not going to undergo any type of differentiation at this point until they see antigen. And so they're going around now looking for antigen. And these T cells can circulate through the body pretty quickly. So when you're looking in the microscope, if you're looking at blood, I'm sure all of you have seen a blood smear. You've seen all those red blood cells across the board, and you've seen probably some neutrophils, some granulocytes, eosinophils. But when you're looking through that, sometimes you'll see the lymphocytes, and they're not much larger than red blood cells. And the only thing that differentiates them pretty much between the red blood cell and them is they're going to be a little bit larger and have that nucleus. And so if you think about it functionally speaking, they're very small round cells, which allows them to quickly and easily circulate through the body as opposed to larger eosinophils and granulocytes and whatnot. So these circulate through the body every 24 hours or so. They'll go through your circulatory system and then through the lymphatics and make that circuit over and over again, looking for their cognate MHC antigen. So as they're looking around, and if they finally find antigen, if they finally find a cell that's expressing the MHC antigen combination that they recognize, then they are going to do one of a few things. And so this is where, in general, just again a reminder, T cells require antigen presentation as the first signal, but then, and then also a reminder is, as opposed to antibodies, that T cell receptor that recognizes MHC and antigen, that binding, that affinity isn't as strong as an antibody, and so it requires a lot more than just one antibody to be bound or one antibody receptor to be bound. You have to have a few MHC molecules that have antigen, and they're recognized by a handful at least of T cell receptors. And then you also have to stabilize that interaction in order to get an activated T cell. So we'll talk about that. Other molecular interactions can provide second required activation signals. So not only do they need that primary signal of seeing antigen, but they need to also receive another signal that is kind of the okay to go ahead and proliferate. It's telling them that, yeah, this is not a self-antigen. So that other signal is going to be co-stimulation. And then once they receive those two signals, they can differentiate into different forms. They can become effector cells, memory cells, but then they can even become different subsets of type of helper cells. Or they can even become regulatory cells and suppress the immune response. So depending on the cytokines that they see now, so there's really three signals we have to keep in mind. There's that primary signal of their T cell receptors seeing that antigen, but then there's going to be, do I see co-stimulation? And there's only certain cells in the body that have those co-stimulatory molecules, so it's not all the cells that are going to be able to induce that. And then also they need to decide, well, what am I seeing in my environment? What kind of cytokines are there here so that I know how to respond? Am I going to become what's called a Th1 cell? So am I going to basically help cytotoxic T cells? Or am I going to be a Th2 helper cell? Am I going to help more of an antibody response to antigen, to bacteria, things like that? So we might have one response in response to viruses, and one response in response to bacteria, different response to worms, fungi have a different response. So all these different responses are going to help us tailor that adaptive, and that also helps us to remember that this is part of the adaptive immune response. So it's going to adapt to specific, not only specific antigens, but then tailor the type of response we have based on, well, what is it? Is it a virus, which is inside the cell? So if I have a virus that's inside the cell, it's not going to do me a whole lot of good to make lots of antibodies, because those antibodies aren't going to see their target within the cell. So we might want to help cytotoxic T cells that are going to be able to recognize viral products that are being broken down within the cell and presented via MHC. So we may want to tailor our immune response based on certain factors. So we'll talk about how do our cells know who's who. So again, our T cells are circulating around looking for antigen. And best case scenario, they're going to find antigen being presented by professional antigen presenting cells. So let me put on my laser pointer again. So we've got our professional antigen presenting cells, and here we have our classic depiction of a dendritic cell, but these antigen presenting cells can be B cells, which is kind of a cool thing because B cells will go around also, and if a B cell sees its antigen, so if a B cell sees a big chunk of bacteria and it becomes activated, it sees its B cell receptor now binds to that big bacteria, it will now start to make co-stimulatory molecules, and it will also be a good APC to induce T cells. And those T cells then will help to induce an immune response and help that B cell also. So they work in conjunction with each other. So again, the T cells will come along, and here we see our naive CD4 T cell, our naive CD8 T cell. And if they receive the proper signals, which is going to be their cognate antigen, MHC binding to their T cell receptor, but then also receive co-stimulation. And this is where APCs are special in that they are those cells that express co-stimulatory molecules that give that second signal. So if this T cell comes along and it finds an epithelial cell that it binds to, that epithelial cell is not going to have that second signal, and therefore it's actually going to induce what's called anergy, which is a state in which that T cell now is not going to proliferate in response to antigen. So let's first look at how we activate cells, these T cells. So they see antigen being presented by a dendritic cell. They get their primary signal through their T cell receptor. They get that secondary signal, and now they are also receiving cytokines, because then they start to secrete cytokines. Maybe the CD8 also secretes cytokines. Maybe the APC also secretes cytokines, and those cytokines are going to help direct those cells to become either helper cells, memory cells, effector cells. So they'll go and they'll populate, because you want certain cells to go out and help or kill cells that are infected, but you also want cells to kind of populate the periphery that have memory, that are going to hang out and keep an eye out for if we ever see this pathogen again, we want to mount a quick response. So we have memory cells also induced. And then looking in on what exactly is happening when a cell sees, when that T cell binds to that dendritic cell. Again, those MHC interactions with T cell receptor are fairly weak, lower affinity than antibody. And so, in order to help that along, we basically, when the cells get together and they see, and you can see these forming when they've done some cool videos where they can actually see the cytoskeleton of the responding dendritic cell once it emanates. So the T cell comes and it binds to that dendritic cell. The cytoskeletal, so those filaments start to drive more MHC molecules to the surface in the location of where that T cell receptor bound. It starts to form, co-localize all of these MHC molecules in what's called the CSMAC, or the central supramolecular activating complex. And so you get this CSMAC, which has all of your MHC molecules, all of your T cell receptors will form in this central region. Also your co-stimulatory molecules, your CD28 and your CD80s. So on your T cell, you have CD28 as the receptor, the co-stimulatory receptor, and that's an important one to know. And that receptor will bind to CD80 or CD86. And that is also within that central area. But then to stabilize that interaction and increase the avidity, we also have adhesion molecules like the LFA3 and CD2, or LFA1 and ICAM1. So we have these adhesion molecules, yeah.
So where's the C-SMAC and the T-SMAC on the cell? Like how are they separated?
So they're separated by actual location. So so what we're looking at is if you have a cell, so let's say, I should cross the cell. You have a cell here, and you have your T cell, and then it binds to, so where they touched, right there at that central location, all of the cytoskeleton within the cells actually drives molecules to the cell surface. You get lipid rafts forming, and basically where the two cells touch, right there is going to be that C-SMAC. And all of those molecules are being directed, but then in the periphery of that, you have all these adhesion molecules. And that kind of stabilizes that formation, because the longer that T cell is bound to that antigen presenting cell, the better chance it has of becoming activated. And so people have shown that if you get these cells in culture and you allow them to bind together, the longer that they stay stuck together is the better chance you have of forming an effector cell, as opposed to getting a cell that becomes emerging. So based on the length of that time and how long they can communicate with each other, that plays a role into how they differentiate as well. We're not going to really get into a whole lot of that, but that does play a role. So they do want to stabilize, and that's why you have those adhesion molecules that help to strengthen that synapse between the two cells for a period of time, so that they can communicate, secrete cytokines, receive co-stimulation, and then become an effector cell. So, co-stimulatory signals are required for optimal T cell activation. You've got your signal 1 again is that antigen-specific T cell receptor engagement. Signal 2 is contact with co-stimulatory ligands, and then that signal 3 would be cytokines directing T cell differentiation. So this is just a reminder of those 3 signals that are required. So what are some of those co-stimulatory molecules? And this is here where the first one that we discovered was CD80, and its receptor on the T cell receptor is CD28. And so here CD28 is expressed on almost all T cells constitutively, so they're always going to have this expressed. The majority of all CD4s express it. In humans, like half of our CD8s express in mice, all T cells in mice express it. So nobody's really clear as to why only half of our cytotoxic T cells express it. But CD28, so just think of it as constitutive, always expressed. And so these cells are always, these naive cells are always going to have this receptor looking for its co-stimulatory molecule, and in this case it could be CD80, it could be CD86, and that depends on the cells. So for example, like CD80 is expressed constitutively on dendritic cells. Again, dendritic cells are, when we're talking about antigen-presenting cells, the dendritic cell is going to be your number one player. But also B cells can upregulate CD86 when they become active. Macrophages can also upregulate. So if a macrophage is in an inflammatory environment, it will take up antigen. If it sees certain signals, danger signals, or pathogen-associated antigens will bind to that, to the receptors. The PAMPs, remember, or DAMPs are also damage-associated molecules. So pathogen-associated molecules, damage-associated molecules will activate cells, macrophages, they'll activate B cells, and then they will upregulate. So dendritic cells constitutively have these co-stimulatory molecules, whereas like B cells and macrophages are other, they're APCs as well, but they have to upregulate these molecules. But once they do, they can bind and stimulate. We've discovered other in this list, we're just showing you two, but there's at least 10 or more, probably even more, and we're discovering more and more co-stimulatory molecules. And why do we have all these different kinds of co-stimulatory molecules? If we just compare CD28 and ICOS, ICOS is one of the, ICOS isn't constitutively expressed, but so mature cells will upregulate ICOS, and so what people have, researchers have kind of conjectured that ICOS is involved in maintaining memory. It's involved in more long-term, trying to maintain a long-term T cell activation, because what happens with T cells too is in order to prevent cytotoxic toxic shock syndromes and just the T cell in autoimmune diseases. Once T cells become activated, shortly after that, about 24 hours, they start to upregulate these negative co-stimulatory molecules. CTLA4 is one of the first ones that we discovered. So CTLA4 is closely related to CD28, or actually, and it looks very similar, but it signals differently. And so instead, and it actually doesn't signal, so what CTLA does is it basically competes with CD28 for binding of CD80. So if you can look here, CD80 binds to CD28, but it also binds to CTLA4. So what happens is T cells, once they become active, after a period of time, will start to make more and more of this CTLA4. So that peaks at around 3 days after activation. And so about 3 days after that T cell got activated, it's making lots of CTLA4. And CTLA4 binds much more strongly to CD80 than CD28 does, and it outcompetes and basically will cause the cell to stop getting that signal. It will induce that cell to become more of an inert cell. So the state of energy can be kind of turned on and off as well with T cells. And so some people who have tried to treat, for example, cancer with T cells that are specific for those tumors will also give anti-CTLA4. So the anti-CTLA4, because it will block CTLA4 from binding, will allow those T cells to continue to proliferate in response to tumor and give a longer response. PD-1 is also one that people have discovered. It's also a negative co-stimulatory molecule. So that's upregulated again after the T cells become stimulated and activated and becomes an effector cell. It will start to upregulate PD-1. And tumor cells have kind of, we use the term evolved to express PD-L1, the ligand, and by doing so, they can create an environment that is trying to put the brakes on T cell activation. And so this is another thing that we evolved to have these negative co-stimulatory molecules, in large part to prevent autoimmune disease, to prevent too much T cell activation, because we don't want to let them just go forever. But then other systems and bacteria and viruses have learned to induce these negative co-stimulatory molecules. So that's important to know and understand. And a lot of researchers have dedicated their lives to studying these and trying to learn how these manipulate the immune response. So a lot of therapies target either trying to stimulate CD28 or try to block things like CTLA4. So there are a lot of antibody therapies out there that target these co-stimulatory molecules. So again, CD28, this 44 kilodalton glycoprotein homodimer expressed by the majority of T cells. It enhances T cell receptor-induced proliferation and survival, binds to B7 and B7.2 or CD80 and CD86, and it's expressed by antigen-presenting cells. Again, dendritic cells will express those constitutively, but other cells like B cells and macrophages will upregulate them. ICOS is so the important thing again to remember with ICOS is it's not expressed at first, but it is inducible, and it will be upregulated on activated T cells. Again, the negative, and this is just again covering kind of what I talked about. Induced within 24 hours, peaks around 2 to 3 days. CTLA4, it binds to B7 and B7.1 and 2 with higher affinity than CD28, but it shuts down that signaling pathway. Okay, it puts the brakes on. PD1, so PD1, this is, again, this probably wasn't in textbooks a couple of years ago, so PD1 has been a new thing, but we found PD-L1 is expressed on lots of tumor lines, lots of tumors that are untreatable. It's and so it's a target for immune therapies is to block that PD-1, PD-L1 interaction. So what happens if cells do not receive that second signal? And so Carl June, the same guy who invented those CARs, those chimeric antigen receptors, the T cell, B cell receptor chimers, his group identified that you need that second signal. They saw that you can take T cells, and if you stimulate them with like anti-CD3, so you just activate them regardless of what they're bound to, that wasn't enough, because that would actually induce them to now be unresponsive, and you would get cells, they would not proliferate. They would then not respond well to antigen afterwards. But then if they were to have antigen-presenting cells in the plate with those T cells, then they would get cells that could be stimulated and look like good effector cells. And so they started to try and see, well, what is the signal? What is unique about these dendritic cells? And they started playing with antibodies, and they could block. They found antibodies that blocked anti-CD28, and so if they blocked anti-CD28 from binding to CD80, then they would, or if they were able to block that, they wouldn't get effector cells. But if they were able to stimulate through that CD28, they would get effector cells. So they're able to do a couple things. They were either would have APCs with no co-stimulatory molecules, and those would induce anergy, or they would block with antibodies in that way. So they were able to identify, in the first one they identified with CD8.
When you say
knock the gene out. With those.
So the first one is knockdown,
Mhm.
the second one is normal.
Yeah.
But why they
And then
gonna,
this one, they blocked the binding between the two with an antibody.
but if that one is normal, why do they,
Yeah.
the second one, the B, why they have no response? Because they don't have a second signal or something?
Oh, when they're talking about response here, so I have to go back and look at this experiment. I think they're talking about the second response here, for example. If you have that primary response, if you don't have co-stimulation,
Yeah.
and then you take those cells and then you try and stimulate them, even if you have co-stimulatory molecules at that point, they won't.
Oh, because they're already.
Yeah.
So
Yeah.
once
So that type of energy is non-reversible.
So those, if they don't have considered they die somehow.
They
Die
don't
later.
necessarily die. They are more short-lived, so they eventually do die, but because they're not receiving.
But when their time is not dying, so then you agree stimulate them.
Yeah, and they just don't. They don't They don't secrete cytokines. They don't proliferate. So those are the two things that tell us that we have an effector cells. One, we can look and see what cytokines. So when you do this in a dish, you'll let them respond, and then you collect the supernate, the non-cell portion of the media, and you'll look for cytokines. And if you see a good cytokine response, then you know that those cells became activated. Or if you look at proliferation, they'll look at whether the cells proliferated. So those are kind of the two key elements to tell us if we have an effector cell. So they discovered that you needed that second signal. So then the third signal now is cytokine. So initially, the first cytokine that people really discovered and understood was IL-2 because it was so important for proliferation. Depending on what cytokines are present, T cells become activated, different outcomes can occur. IL-2 is the example of an autocrine type of cytokine. So it's, again, once cells become activated, they start to secrete more IL-2, but they also upregulate the high affinity IL-2 receptors, so they make you see CD25 on their cell surface. CD25 is the receptor for IL-2, so not only is the same cell making the cytokine, but it's also making the receptor for the cytokine, and you get this loop of activation, and then other cells around it will also bind to the IL-2, upregulate the IL-2 receptor, and then they'll start to proliferate and secrete IL-2, and so you get this kind of domino effect. Other cytokines can send the T cell down different subset pathways. So we have, how does that happen? So we have antigen presenting cells that have different co-stimulatory properties. Again, here's just an example of our three major subsets of antigen presenting cells. We have our dendritic cells, we have our macrophage, and our B lymphocytes. And so our dendritic cells, once they see signals, will upregulate more MHC class 2. It shows that here's showing a picture of co-stimulatory molecules. So even though they're constitutively expressed, they're upregulated, and so they'll secrete more and more of them. Same thing with macrophages. Once those PAMPs or pathogen-associated molecules are expressed and seen on the surface, then these cells will start to upregulate co-stimulatory molecules, class 2 and class 1. And then B cells are interesting in that when a B cell sees an antigen, so it sees this large chunk of protein that is on a bacteria or something, it's on a worm. So those B cells will bind to that, and then that binding will induce that B cell to become activated. That B cell will now internalize, if it can, that antigen and present it via class 2, and then it will become an antigen-presenting cell. So in these cases, these guys are just presenting whatever antigen is in their area. With the B cell is actually presenting a representation of the antigen it recognized. And so it starts to present lots and lots of chunks of that antigen via MHC to T cells. Now, the T cells also recognize that as foreign because they bind to it. They'll start secreting cytokines, and they'll actually become Th2 cells. They'll start to help secrete cytokines that will help that B cell become mature and start to proliferate, become a plasma cell, start making lots and lots of antibodies. If that B cell doesn't get that help, a lot of times those B cells will just become emergent. So again, if you're developing a vaccine, you might say, well, I'm going to develop a vaccine and I'm going to use this protein that I know is expressed on this bacteria. But you may want to also keep in mind that you may be trying to induce a B cell response, but you might also want to make sure that there's an epitope within that protein that is presented via MHC, and that can also induce a CD4 helper response that will help promote that antibody production. And so that all comes into play with a good vaccine that works well. Is a lot of times that they'll have a target that, and that also comes into play with vaccines. Tetanus is a good example. We still to this day will give people a tetanus shot, and that tetanus shot includes a whole protein. You can use, there are tetanus shots that have a small chunk of protein that's recognized by B cells, but the actual whole protein is much more effective, creates much better memory, is more effective in a larger population. So if you take a smaller chunk of that protein, and people started to do that because they're afraid that, because the protein we're using for tetanus shots is actually a pathogenic molecule, and so it can do damage if it's not attenuated. But it also doesn't work as well as a vaccine if you don't use the whole thing. So that probably comes into play in that the more possible epitopes you give, the better help you're going to get from T cells. So always things to keep in mind when you're trying to develop a vaccine or use a vaccine.
But you don't know which chunk they are going to have an actual then.
Well, and it depends on the MHC of the patient as well. So we all have different MHC, so we're all going to present different chunks, different pieces of that protein via our MHC. So you're going to present. So that's why if I give a smaller chunk of the protein, then I am limiting the percentage of the population that's going to effectively induce a helper response. And so by providing the whole protein, I have a better chance that your MHC is going to present one chunk and this person's MHC is going to present a different chunk. But they're both going to give help to that B cell to see a larger chunk. So it's all.
But to say that it's easy, but when you have tetanus shot, it's very painful.
It is painful,
Even
yeah.
for a small piece.
Yeah.
How about a bigger piece? Do I feel awful?
You know, that may play some role.
Because I had to take it every 10 years.
Yeah.
Or 5 years.
Yeah. But and actually, the worse the immune
For 3
response
days,
is, the
I'm
more sore you're going to be.
cranky.
Yeah, those tetanus shots are not fun. But so the other thing to keep in mind too is we have these things called super antigens. We only have one slide because it's really not. But what a super antigen is important to know is a super antigen bypasses a T cell receptor specificity for a certain MHC. Super antigen binds to the variable region, the B beta, but it binds to all B betas within a certain class. And it binds to that B beta, so it's like as if I had an antibody that kind of stuck these two together. And regardless of whether he recognizes this antigen, this peptide, this is going to cause them to bind together as if they did recognize each other. And so if certain bacteria express super antigens, food poisonings caused by certain superantigens. I think botulinum toxin, whatever that bacteria is, it has a superantigen. So superantigens, what they do is they basically will bind non-specifically to T cells and activate them. These T cells then will start to proliferate. And people, you might ask, well, why would a bacteria try and induce a T cell response? But what's happening is it's inducing a T cell response that's not specific to it. It's inducing this huge array of T cells that get activated non-specifically. They start to secrete lots and lots of cytokines. They suck up all the cytokines in the area, and it kind of is like confusing the immune system. So you can't really induce any type of specific immune response. And a lot of researchers use these superantigens as ways to really activate T cells in culture. It's a tool that we'll use to non-specifically activate T cells. And it will activate all T cells with a certain V beta. So when I say a certain V beta, let's say 5% of your T cells will have a given V beta that's recognized by the superantigen. And so basically you're turning on just 5% of your T cells, not specifically. So superantigens are expressed, and some of them, the ones are SEA, SEB, and these are food poisoning, toxic shock syndrome. So these are expressed, and so these are sometimes what it's these superantigens that are very dangerous. It's not necessarily the bacteria that gets you. So that's why even if you've had a can, let's say you have a can of food and you see the bulging, so it's got botulism in there. Even if I take that and I was to boil that, I know there's no bacteria in it. I killed all the bacteria. You still don't want to eat it because it's got that superantigen in there. That superantigen doesn't get denatured. They're pretty stable. And so I eat that can thinking it's safe because I killed all the bacteria, but you're still going to get this huge cytokine response you're going to get. You're basically going to feel like you have the flu times 10. You're going to be, your temperature is going to go up. You're going to have this huge cascade. Hopefully you survive, but many people don't. So that's what's dangerous about those pathogens.
So, well, I was just gonna ask, addressing, so how do you choose a superantigen?
It's pretty hard to denature.
Like bleach or something?
Yeah, but bleach pretty much will denature a lot of stuff.
Oh, you cannot.
But you don't really want to. Yeah, you don't want to. Yeah.
Well, I'm not talking about foods.
So yeah, it's if you had an antibody against that superantigen, that might be a way to clear it quickly.
So how do you put a staple couple or imperial? It's only on skin, right? If I understand correctly.
Not all staph, but yeah, strep. So this is it's a certain streptococcal pyogenes exotoxins. Yeah. So can lead to rheumatic fever, shock. Yeah.
How do you cure it?
How do you cure it? Well, first you got to treat it with antibiotics to get rid of the bacteria so that you can limit the amount of superantigens. But then a lot of times a treatment for something where you're having toxic shock would be maybe trying to block the immune response of the cell. And a lot of times,
You get trigger syndrome.
yeah. So that initial activation signal 1 and 2 induces upregulation of pro-survival genes, transcription of IL-2 and the IL-2 receptor genes occurs. So this is all going to occur shortly after the activation, and the outcome is proliferation, production and effector clonal cells. So you get some memory cells, some effector cells. How do we decide? How does a cell decide whether it's going to become a memory or an effector cell? Is a good question that researchers are still trying to figure out. It may just be a numbers game. It may be how long the cell was down, whether it's becoming a memory or effector cell. It may just it may be cytokines. Those cells that got the most cytokines. So it's something that is still under investigation. But over time, the stronger the response and the longer the response, the more memory cells you get. So memory cells don't seem to be dictated on whether the affinity of the T cell receptor. It just seems to be the longer you have. So that might suggest that it's just a numbers game, whereas a few of those cells are going to become memory cells somehow.
What do you mean by that? The stronger, the longer you become more inactive?
The longer you have an immune response, so the more cells that proliferate in response to that antigen, the more memory cells you end up getting.
The longer, not stronger.
Yeah, not stronger. So some people used to initially were playing with ideas. Well, maybe it's based on the type of antigen. So maybe we'll produce memory cells if we have a strong antigen response, whereas maybe we don't produce them when we have a weak one. But that doesn't seem to be the case because when we experimentally
How about
look
the
at
stronger? Is stronger going for the effector?
that was something also people were conjecturing that maybe those cells that became memory cells maybe were bound longer, but there's really not a lot of solid information out there
Yes.
yet.
They don't know how they become.
Yeah. So that's one of those things where we'd like to understand it better so that we can try and induce memory better. Again, you have certain vaccines, for example, that you need a booster at every. It would, if we could induce better memory, maybe we could give fewer vaccines. If we can induce better memory, maybe we wouldn't have to have as many boosters as often, things like that. And that is something that people are really interested in figuring out. So, helper T cells, they can be divided into these subsets and many more. We're just going to talk about these, but they are not limited to these. There are more. So at least, we say at least 5 distinct subsets. Th1s and Th2s were the first ones that people were able to see in vitro. It was harder to find them in vivo because these subsets are usually actively responding, and then again they upregulate negative co-stimulatory molecules like CTLA4, and then they go away. So it's a lot harder to find them in vivo. But so eventually we did start to find these, and so Th1s are going to be your response to viral antigens, and Th2s are going to be your response to bacteria and pathogens that are maybe better treated with antibodies, so B cells. So Th2s will induce B cells, whereas Th1s will induce cytotoxic T cells. And we also have Th17s, which have been shown to be involved in removal of fungal things, but they're also found very highly in people with chronic inflammation, people with chronic autoimmune disorders, Crohn's disease, things like that. You see a lot of Th17 cells. So we may have evolved this subset to give some people think that maybe we evolved to have this subset to deal with things like fungus, which is kind of a long-lived, really hard to get rid of a fungus. And so we couldn't clear, we couldn't clear fungus with the Th1 because this Th1 is kind of this short-lived but very strong immune response to get rid of a virus. But maybe that wasn't sufficient, and we were having cells that were attacking fungal cells, but then they were turning off after about 3 or 4 days, and the fungus was still there, and then it would take off. Th17s seem to be more long-lived, and they will respond over the long term, but then they also now are involved in issues with possibly even inducing chronic inflammation get shut off. They don't have as short-lived a lifespan, so they're more involved in autoimmune disease and also and so people have actually tried to, some researchers,
What do you mean that, TF17, so that people with autoimmune, they have that?
Yes, same.
yeah, they have a large percentage of their cells are Th17 cells. And so that's where people started to kind of
They want to
experiment
kill
with
it,
things.
they want to get rid of it.
Well, in autoimmune disease, you want to get rid of them.
Yeah.
But in the case of a fungal infection, you want them. So, that's the yin and yang to the immune system. And people who have been making tumor vaccines, for example, most people who develop tumor vaccines have always focused on the Th1 subset. So when we're trying to induce T cells that attack something, we said, oh, let Th1s are good at getting rid of viruses. And tumors aren't viruses, but they're presenting endogenous antigens. We want to get rid of these tumor cells. And so most researchers focused on Th1 cells. For the most part, those work really well in vitro, but for in vivo, they work initially, but then they stop working. So, but tumors continue. So maybe you see your tumor load go down for a bit, but then it takes off. Okay, so have actually, a lot of researchers who are trying to develop tumor vaccines have focused on this Th17 population because it is what's involved in trying to remove things like the fungus that are hanging around for longer. So this is a more long-lived immune response. So those cells could be important for tumor immunotherapies, but they may not be so great for, again, autoimmune disease. So in some cases you might want them, in some cases you don't. Regulatory cells. Here's the cells that are important. We talked about, so regulatory cells can be made in the thymus, and go out into the periphery and suppress immune responses against self-antigen. But if a T cell gets out of the thymus, and it's specific for self-antigen, we can also induce Tregs. So they're called iTregs, inducible Tregs. So we can actually, if a CD4 cell sees antigen, but there's no danger signals, it doesn't see it in the right context, certain cytokines will drive a regulatory response. So chronic inflammation can actually drive more Tregs. And so these Tregs will then suppress other T cells. And so these are going to be cells that are involved in helping to prevent autoimmune disease, these Tregs.
When you say Treg, is with the common one and the inducible one?
So now we're talking about inducible ones. So in these, so Tregs, and actually Th17, some of the signals that will produce a Th17 cell will also produce a Treg. And Th17 cells can become Tregs as well. So it's kind of a fine line in between these two guys. And then we have these T follicular helper cells. And these are a subset of cells that were recently discovered of T cells that help to induce B cell responses in the follicles, lymph nodes, and the secondary lymphoid organs. So these are important in B cell help. So each produce distinct. So that was kind of just the kind of going over what each is kind of involved in. How do they decide which one to become? It's that third signal that helps them decide what they're going to become. Those cytokines. So what cytokines do they see? So APCs may bind to PAMPs via their PRRs we talked about earlier on chapter 3, I believe it was. And these induce cytokine secretion. So depending on the signals that the dendritic cells get or the APCs get, they're going to start to secrete cytokines themselves. Those different PRRs engage these different antigens and different cytokines. So viruses will stimulate IL-12 to produce a Th1 subset, whereas worms, so worms aren't going to be treated as well with, worms aren't going to be treated very well at all with cytotoxic T cells. Because well, so does a worm have MHC molecules on it? Probably not. That would be really weird if it had human MHC molecules on it. So unless that worm gets endophagocytosed, broken down into little pieces, and then presented via an antigen presenting cell, we're not going to be able to induce.
So worm are not,
So-
cell.
So we want to attack worms with antibodies, right? So in order to get rid of worms, we want to induce B cells. So even though we got a helper cell, maybe we killed a few worms, maybe we phagocytosed one worm and presented its antigens to this helper cell. Those worms that are still out there, floating around, swimming around, getting into stuff, we want to get rid of those guys that are doing damage, and so we're going to do that best with B cells. So worms will induce a Th2 response. So we don't want to focus our efforts on cytotoxic T cells. We want to focus our efforts, because in general, worms aren't infecting cells; they're eating cells or damaging cells.
So, I still don't get, why the worm you attack the TH2? Because it's similar,
So helper cells are going to help B cells, right?
Yeah.
We don't need cytotoxic T cells because we don't have cells that are infected that need to be killed. We have a whole worm that we need to kill, and we're not going to be able to kill it with a cytotoxic T cell.
So that means no cell is infected with it.
The worm. Those cells are infected with the worm. These worms are big and they're out there and they're eating our food and they're hanging out in our apartment and we just want to get rid of them. And so we won't be cells. Okay, so some of the cytokines involved in those different responses, depending on if you see a viral antigen, bacterial antigen, worm. So these are things associated with worms that will bind to a receptor on an APC, and that will make the APC secrete certain cytokines. So some of the cytokines, for example, IL-4, so it's important to memorize, IL-4 will induce a Th2 response, but that is that major cytokine in driving that Th2, driving a B cell response. So if you can just remember Th2, IL-4, B cell response, that's going to get you a long way.
Yeah, can you, what did you say?
Th2, so Th2 helper cells are induced by IL-4, and those are going to help B cells. Those are going to drive B cell proliferation. Th1s are going to be induced by IL-12, interferon gamma, IL-18 as well, but
I just remember IL-12, that's it?
yeah, interferon gamma is a really important one too. So these are going to induce, again, interferon gamma, remember, has antiviral activity itself. And so in response to viruses, cells produce interferon gamma. So if you can remember all the way back to when we were talking about that, that interferon gamma actually has innate response, is a part of the innate response against the virus. Interferon gamma itself can induce antiviral activity within a cell. Interferon gamma. So, and in the same vein, interferon gamma will, if it's secreted in an environment, it will upregulate co-stimulatory molecules on APCs. Interferon gamma will also then drive a Th1 response, which will then drive cytotoxic T cells to now. So think Th1, interferon gamma, IL-12, and that's when we do cytotoxic T cells. Th17 is going to be induced by, here we have TGF-beta, but if you look down at Tregs, TGF-beta is also there. IL-6 is going to be the decider. If you have IL-6 with TGF-beta, you get a Th17. If you have TGF-beta and the proper signal, IL-2 signal, you get a Treg. And what makes a cell a Treg versus, so we have oversimplified things, but just for the ability to kind of teach this, Tregs can be induced. It's not 100% understood. Treg induction is not 100% understood, and over time you do get more and more Tregs, and so it seems like a small percentage of effector cells, of helpers Th1 cells, can actually may not want to induce regulatory cells if we want to induce an immune response. And we've seen that now in tumors, and when we give tumor vaccines, if we give them quite often, we get developing lots and lots of Tregs. Now that could either be the tumors are secreting lots of TGF-beta, which is the case in some cases, but it could also be that eventually you do develop over time. The more and more you see an antigen, the more and more Tregs develop, and you eventually get just too many Tregs. More Tregs suppressing the immune response than you can get vectors. And that's possibly why also when people with cancer are given they are given chemotherapy that removes all of the T cells. It also removes the Tregs, and that when you then repopulate their system with just naive cells, oftentimes you can get a really robust immune response against those tumors because you've eliminated that peripheral tolerance that's induced over time, that buildup in Tregs. And then we've got our T follicular cells. So again, IL-6, IL-21, and some of these cross over, but the combination of them will drive that one population. So the master gene regulators,
We only have one day.
Oh, sorry.
yeah, both not going to be like that, but
Yeah.
Yeah, I really wouldn't focus on memorizing those, just because of my personal background and interest. I would say that it's important to know that FOXP3 is the master gene regulator for regulatory cells. That really is really important. That's how they kind of identified who's a regulatory cell versus not. And it really is the only way to identify truly identify a regulatory cell.
So why? Because they all have.
Well, Th1s and Th2s are actually can be identified without identifying Tbet. For example, you can show by just certain receptors that are expressed what's Th1 cell. So I can with just antibodies on the cell surface, I can tell who's the Th1 versus the Th2. Even the Th17, you have a IL-17 receptor. Tells me that's a Th17. All of these cells except for the Treg can be identified by surface markers. The Treg looks just like a Th1 cell, except it expresses FOXP3. And so, if you really want to identify Treg, you have to stain intracellularly for that transcription factor for FOXP3.
So you mean you use it for immunofluorescence?
Yep. So you actually have to kill the cells, which is why studying Tregs has been so difficult in the past because, because people researchers are desperately looking for a surface marker that identifies a Treg versus an effector cell, and they haven't really found a really good one yet.
So they didn't have to flow cytometry?
They can do flow cytometry, but you have to punch holes in the cell to get the antibody in to stain FOXP3, or you have to make a cell. You have to make a mouse, so we can't do it. You have to make a mouse that expresses green fluorescent protein driven by the same promoter for FOXP3. So only Tregs would be green in that case. So you can study it in vivo with like mice, but it's really hard to study in humans because you have to kill the cell, and once you kill the cell, now how do I study its activation? How do I study what it does, what it's doing? And so some people have argued that inducible Tregs Tregs are actually reversible. But it's hard to really show that because once I've stained a cell and killed it, I can't then put it back in culture and see if it goes back to not being a Treg. So it's really a hard thing to study. And people have seen that Tregs can be induced by so Th1 cells can become Tregs, but then some people argue that maybe they can go back. So they just might become Tregs just like cells make CTLA-4 after 3 days. They might become Tregs to suppress the immune response, but then they'll convert back to effector cells once, after a certain period of time. So that big no need to know. But that is.
Before you go, sir, so for this polarizing cytokine, right? So they have major one, like for instance, for Th1, whatever, you just remember it's IL-2, that's okay, right? No, that's Treg.
Yeah.
You heard
Well, IL-2 is also for Th1, but again, that's going to be combined with other cytokines. And sometimes if I have a dish, let's say I put all these T cells in a dish, I'm either going to get Th1 cells or I'm going to get Th2 cells because they're competing on those. They have competing cytokines, and so you're not going to get.
So these cytokine are major one, but they have minor one too, right?
Yeah.
So it's not exclusively just for that.
Yeah, it would be easy if it were just one cytokine driving one immune response. And so that kind of shows the complexity. I mean, if you look, here, I'm just going to try and get out of this in a sec. If you look at, here's I didn't necessarily want to scare anybody, but this is just, and this isn't even the whole list. We've got Th1 cells, we've got Th2 cells, Th9 cells we didn't even talk about, Th17, Th22 we didn't talk about. Here's our follicular cells. We have Tregs, we have natural Tregs, inducible Tregs. So, I mean, and this is just a recent review.
Are we supposed to be saying this?
Do you not see it? Oh, boy.
Good.
There we go.
Oh,
So,
that's good one.
so I'm not going to put this up or anything, but let's see.
Forget it, we don't even know that.
So I mean, you have Th2, but you also have Th1s, Th2s, you have Th9s. I mean, and these are the different cytokines. And this is you've got your Th17s over here somewhere.
Yeah, that
Top. You have your Th22s, your T follicular cells. You have your natural Tregs. These are the ones that come out of the thymus. Already Tregs, and those people think are not. They're going to be Tregs. They're not going to go back to being something else. They're going to stay a Treg. They're always a Treg. And in people who've done studies, that's pretty solid. But these inducible Tregs, these are the ones that are induced in the periphery, and that's the big question as to whether those are stable.
yeah, we only have 10 minutes.
Yeah. So, for the most part, you just need to know kind of the key cytokine and what it's gonna and what are Th1s? They're involved in viral responses and inducing cytotoxic T cells. Th2s are involved in, let's say the example would be worms, in inducing B cells or antibody production. Th17s, not so important to know what they're driving because they're similar to Th1s, but they're responding to more fungal infections, but they're also associated with inflammation, chronic inflammation. T follicular cells are only going to be found in the secondary lymphoid organs, and those are involved in the zones where T cells and B cells are talking and basically helping to induce B cell proliferation. So, and then Tregs are going to be involved in suppressing immune responses in the periphery. And that's based on what signals, what PRRs are the antigen presenting cells themselves getting. So the antigen presenting cells are getting information. They the information to the T cell via what cytokine it secretes. And they believe that even in the T cell is actually, there's current research saying that the T cell is also communicating with the APC and driving certain transcriptional events within the antigen presenting cell itself. So again, effector T cell subsets, for example, the Th1 versus Th2 cross-regulation, the same stimuli that stimulates an IL promotes a Th1 is going to inhibit Th2. And those Th2 cytokines like IL-4 are going to inhibit Th1 response. So this kind of helps strengthen whether I'm going to have a Th1 versus Th2, and this is why you will probably never see both occurring at the same time, because if I have more interferon gamma than I have IL-4, I'm going to get Th1s. If I have more IL-4 than interferon gamma, then I'm going to get a Th2, and it's going to block Th1. So the same thing happens between Th17s and Tregs, where the deciding factor is IL-6. So effector T cell, so this is just going more into what the Th17 cell subset is involved in. You can go over that on your own. Tregs, again, the Treg itself is also stimulated or activated by TGF-beta, just like the IL-17 cell. And these Tregs will secrete suppressive cytokines. They will, and they're going to suppress T cells directly. This is just talking about that Th17 versus Treg cross-regulation, just like we had cross-regulation between Th1 and Th2. And follicular helper cells, this just talks about who they are and what they do, and I already talked about that. And here's just a bullet point also saying that just because we have a Th subset of cells doesn't mean that they can't revert to a different. So there's a lot of research showing that T cells are a little more fluid with what they become. A Th17 might be able to become a Treg or vice versa. So you might get some floating between Th1 and Th2, or between these two, but not necessarily a Th1. That Th1 again, the Th1s look a lot like Tregs. Again, looking at surface markers, but sometimes FOXP3 that can be induced in Th1s, and that will make it become a Treg. And so we used to see this a lot when we would take T cells and you would stimulate them in culture. And you looked at Tregs. The first time, for example, the first time I stimulated cells with tumor, we would have about 5%. So most of us have about 5% Tregs, circulating Tregs. 5% of our CD4s are Tregs. And then you would induce them with the tumor line, and you would get proliferation in effector cells. But then about 10% of the cells would be Tregs. And then you do it again, and about 20% of the cells would be Tregs. And you do it again, and about 50% of the cells would be Tregs. So over time, you can drive expression of, or you can drive differentiation towards Tregs. The question is whether they can come back. So these subsets play a critical role in immune responses. Just go over that. Here's an example of surface protein expression, how we know what's a naive versus effector memory. So we can tell what memory cells are. And when you have memory cells, you have central versus effector memory. So central memory, a lot of times this is just based on the location. Central memory cells are going to be found in the secondary lymphoid organs, whereas effector memory, so those that are going to respond and turn into effector cells, are more out in the periphery. They're more in the tertiary lymphoid organs, the skin and the gut, and those are going to be there ready to respond very quickly. Again, we don't understand a whole lot what makes a cell become an essential memory versus effector memory. Again, that's something that is really key that people are trying to understand. And this is one of those throwaway slides. How and when memory cells arise, we don't know. Okay, that's long story short. What signals induce memory cell commitment? Again, kind of clear. So you don't have to know it. Are there differences between CD4 and CD8 memory cells? Probably. Now we are getting into the realm of things that it's now. There are all these crazy subsets. They obviously have to have some role. We know that they have major roles because if you don't have if you knock out the ability to have Th17, you have people who will be increased risk of getting fungal infections. You know that if you get rid of your ability to have Tregs, or people that have mutations in their FOXP3 gene, those people have autoimmunity, they have chronic inflammation, they have issues. So each of these subsets is really important for what they do. What we're still trying to learn is all the different subsets. How do you induce them? Are they reversible? How do you get regulatory T cells? How do you get memory cells? And again, as you saw, we're just showing you a small percentage of the different cytokines involved. Now, we are discovering cytokines all the time. And so the interplay between these cells is really complicated. But as far as learning it, we try and focus on what really the key players are and what we really do know. And so those are going to be the ones we kind of present. So if you study all that, you'll be ready for the exam. I promise not to throw any crazy questions out there. I've been going through the questions, making sure they're not so obtuse as they have been in the past, and so hopefully these will be straightforward questions for this exam. Again, focus on chapter 9 and 11. Don't forget chapter 10. But I want you to know for chapter 10, I want you to be solid on what the basics are for chapter 10. Okay, know the concepts, understand that. I will try and keep the